Okay, everyone, welcome to another edition of College of Science Virtual Seminar. As usual, I'm still Michael Kaufman. I always say my name. I don't really know why I do that because everyone has heard me say this a million times. We're doing this in hybrid mode today, and it's interesting. We have exactly two of us sitting here watching the seminar in person, Jessica Castillo-Bradaro and myself, and Maya's over there, as you can see. But it looks like we have another 22 people online, which is a great turnout and maybe an indication that people like to do these virtually. So we'll give it a try. Anyway, thanks for joining us. Let me give you a little bit of background on Maya, like I usually do. Maya got her BS in evolution and ecology from UC Davis some time ago. And then went on to do her PhD in integrated biology with a focus on the morphology and ecology of stomatopod crustaceans. I think I got that about right, at UC Berkeley. Once she finished at Berkeley, she was a visiting fellow at the Nautilus Marine Laboratory in Panama, one of many exciting locations it looks like she's been, and I'll mention those in a minute. She was a postdoc at Scripps Oceanography, and then the director of training activities for sustainable oceans for the NSF National Research Traineeship on Coastal and Marine at the Coastal and Marine Studies Institute at UC Davis, before interviewing for a job here at San Jose State University. She interviewed in fall of 18 and delayed her start by a semester to spring of 20, which happened to be the semester when all hell broke loose around here. So she got to be in person with all of us for about 3 months before the place shut down. And she was just saying to me a few minutes that she doesn't think she's done an in-person seminar since joining San Jose State. Would that be correct? Yeah, I think that's right. Just to give you an idea of the exciting places where marine biologists get to go, here's a sampling of the field station experience that Maya has. She has been to the Galapagos, Maui, French Polynesia, the Great Barrier Reef in Australia several times. Let's see, Stadbroke Island, also in Australia, the Panama Tropical Research Institute, and closer to home, Catalina Island. So today, Maya is going to tell us about trophic relationships in the benthos, feeding morphology and ecology of macroinvertebrates. And I had to think for a minute, these really are big for invertebrates. There aren't that many invertebrates that are hold them in your hand, like fill up your hand, right? So very excited to hear about the work she's doing, and take it away, Maya.
I will try this sitting down, but I may stand up for those of you on Zoom. But thank you all so much for coming. I really appreciate it and love these seminars, and I'm really excited to finally give one. So thanks for having me. Yes, as Dean Kaufman mentioned, I work on mantis shrimp, but also a bunch of other critters that I'm going to tell you about today from all the amazing places that I've been lucky enough to go to. And this is a mantis shrimp, and it is about 1 foot long. So this is actually our biggest mantis shrimp species. It's about this big. And it has these amazing predatory appendages that it unfurls and captures fast-moving prey like fish and other crustaceans that are flying, swimming over its burrow. And so it lunges out of its sandy burrow and captures prey. And I am really excited about studying mantis shrimp in the context of feeding morphology and ecology, because they have these like really specialized feeding appendages that they use to do a bunch of things. And that allows us to understand kind of the evolution of how animals feed, what they feed on, why they feed on certain things, because everything in the world has to acquire energy from, well, everything living in the world has to acquire energy from something. And so feeding morphology can really drive the amazing biodiversity we see on Earth. And that's why I'm really excited about this topic. So mantis shrimp, a lot of them live in these beautiful coral reef habitats. If you were to put on your mask and snorkel and go diving here, you would see, of course, all these amazing colorful fish and corals, but you would also see a bunch of kind of tunnels and crevices. And mantis shrimp are poking their little eyes out of many of these. And of course, this environment is just this huge, coral reefs are huge three-dimensional structures, again, with many tunnels and crevices. And they are all susceptible to the oceanographic patterns that we see on Earth. So nutrient flow, light availability, and then of course, in the context of global climate change, coral reefs are especially susceptible to ocean warming and ocean acidification. And so I'm really interested in how animals feed on and defend themselves from feeding attacks, and trophic dynamics in general. That means food web dynamics, so who eats whom, and then how environment relates to all of those things. And I look at that in coral reefs, of course, but also kelp forests. So we're going to talk mostly about coral reefs today, but we will talk a little bit about kelp forests as well, and the exciting kelp forest work that I've been able to start here at San Jose State. So this interplay between feeding interactions influencing food web dynamics and environment influencing food web dynamics leads to kind of the two questions that guide my work. And that's how do morphological feeding specializations and predator defenses contribute to trophic dynamics and ecosystem function, but then also how do environmental disturbances influence trophic relationships? And again, looking at those primarily in these two exemplar habitats, coral reefs and kelp forests. But I want to first start by kind of looking at the broader picture of why I think feeding interactions are so important beyond that, you know, everything needs energy to survive. But also there's this really kind of fundamental tenet in evolution and ecology that morphological specialization, so how specialized your body is, leads to a narrow diet. And the classic example of this are the Galapagos finches. So probably many of you have heard of or seen Galapagos finches. They have these amazing beak morphologies that they use to either crush seeds, like small seeds, or large seeds, like the small groundfinch, the medium groundfinch, and the large groundfinch. But then they also eat cactus flowers and insects from trees, and their beaks are highly specialized to do this. But then, if we look at just kind of the small, medium, and large groundfinches, we know from amazing work in the late 70s, early 80s by Peter and Rosemary Grant, in the Galapagos, that environment really drives morphological changes. And so here is kind of the classic study that they did, where they looked at the distribution of beak sizes before a massive drought and after a massive drought that happened in the late 70s in the Galapagos Islands. And basically you see a shift in the distribution of beak size from small to large. So the beak sizes become larger because basically all of the finches could eat the small seeds, but only the large groundfinches could eat the large ones. And so you have kind of selection towards the large beak size. And so environment really changes and can influence the morphology that we see in the environment. And so another animal that's often touted as having highly specialized morphology are stomatopod crustaceans, or mantis shrimp. Great pronunciation. So yes, there are 500 plus known species. They're found tropically and subtropically, and they have highly specialized feeding ecology again. They've become really famous in the past. Yeah, I mean, since I started working on them in the early, well, like mid to, 2005, I guess I started working on them. Nobody knew what they were in 2005, but now I ask a room full of people. In fact, you all can raise your hands if you want virtually and here. But yeah, if you've heard of mantis shrimp, raise your hand. Now, like almost everyone knows what they are. I have 2 participants, 3 participants. Awesome. 5, 6. Okay, this is looking much better. Yes, yeah. So most people know what they are. They've become very popular in pop culture and media. And here is one of my favorite examples of mantis shrimp in the media. This is a comic by the Oatmeal, and you see this mantis shrimp like smashing the claw off of this crab saying, and now your dinner. And so it's this raptorial appendage that's highly specialized. Here you can see it down here in this animal as well. And it's in all of these. And also I should say that these mantis shrimp are scaled to size. So you have really, really small ones and really big ones like we were talking about earlier. So I actually have a video of the mantis shrimp doing just the thing I showed you. So it's going to, here's its little hammer. Oops, sorry. Here's its hammer, and it's going to smash the claw off of that crab. It just did it. So this is the high-speed video. It's filmed at about 5,000 frames per second, slowed down so you can see it. Again, it's going to rotate forward, smash the claw off of this crab, and the claw goes flying across the tank. So this is one of the fastest movements ever recorded in the animal kingdom. It's going about 23 meters per second. That's about 50 miles an hour, this little hammer here. And so I'm really excited about, of course, oh yes,
So
yes.
that was slowed down, right?
Slowed down, yes.
So what's the time scale? Like, how long does it go take from so to hitting the other crowd? Yeah,
great question. So how long does it take to go from kind of a cocked appendage to a released appendage? So the strike speed, the average is about 3 to 5 meters per second for that species, but of course it varies.
Okay, so that's the speed, but
I'm sorry, milliseconds, 3 to 5 milliseconds, right. 23 meters per second.
Right. So you think about that.
Yes. Yes. Yeah, 3 to 5 milliseconds. Yes.
Right.
And then
stopping again.
Yeah, well, it usually rotates kind of past, but yeah, its appendage does not fly off its arm, which is actually amazing in and of itself too. Yes, there is a whole set of amazing research studies about that I won't go into, but would love to talk more about because it is amazing how they don't lose their appendage basically. Yes, right. So we have the cool morphology, which I'll talk about a little bit. And then of course the feeding interaction. So what is this predator-prey dynamic? What's happening to the prey item? What's happening to the predator? And so, but of course, this feeding interaction is just one feeding interaction among a huge web of a bunch of animals eating each other. And of course, that happens in an environment. So that's happening in this case in a coral reef. So we can look at these dynamics between feeding interactions on the individual level and scale it to the trophic dynamic level and the environment level. So here are a couple more videos. So this is, oops, I want this muted. Oh, well. This is filmed at 30 frames per second. I'm not seeing it on my thing. So this is real time. So you can see what a smash looks like in real time. And then here is the appendage rotation again. So here's the strike again, slowed down so you can see it. There was one flash of light upon impact, and there's going to be another right here, right there. So yes, a little bit later, there's 2 flashes of light, one on impact, one about 11 seconds later, and that's cavitation. So to tell you even more how like amazing this appendage is, basically the appendage is moving so fast through water that it creates an area of low pressure that forms an air bubble. When that air bubble implodes, it releases energy in the form of light and heat that's comparable to the sun, and that's cavitation. Yes, and it's there's an analogy that I've heard that I really like. If a mantis shrimp were human size, and if it were to throw a baseball with the same kinematics that it's using here, it would launch that baseball into orbit around the Earth. So good thing they're not human sized. Yes, and so so this is another species. This is a spearer. So this is that big animal that you saw on the title slide. Here it's going to capture the shrimp on a stick in real time. Showed a couple times. And here it is slowed down again. So this is filmed at 3,000 frames per second. Again, slowed down so that this movement is much slower. As you can see, the linear velocity is like 1/10 of the speed of the smasher, and the linear acceleration is like magnitude slower. But basically, the appendage is doing a different movement. It's opening up, and then it's rotating forward and capturing the shrimp on a stick between these two parts of the appendage. So the behavior is different, but also this animal isn't capable of producing as fast of strikes. And that's to do with the spring elements in its appendage. So both of these animals produce these fast movements using a spring and a latch system. It's basically like a bow and arrow metaphor. So, you pull really hard on the bow, and then when you release it, the arrow shoots forward. That's essentially what's happening here, except the appendage stays on the animal. And there's a whole bunch of, again, really cool mechanics associated with how this works and why these animals aren't losing their appendage. But I am more excited about what they're eating. So I basically have just told you that smashers are super fast and powerful, and spearers are fast and far reaching. So they can reach really far to grab evasive prey swimming over their burrows. Generally, we think that smashers eat hard-shelled prey, so they produce these really high forces because they're producing high accelerations that yield high force, and that allows them to like break snail shells and eat hermit crabs and do things that are generally hard for other animals. And then we think that spearers eat fast and eat soft-bodied evasive prey, like I was showing you before. And generally, we see smashers in hard substrate habitats and spearers in soft substrate habitats. So I'm really interested in the smasher and what it eats. And hopefully I've convinced you now that smashers have highly specialized morphology for producing these amazing movements. And so the question is, do they have a narrow diet of only hard-shelled prey or a wide diet that includes soft-bodied prey? And I've used stable isotope analyses of diet to understand what animals are eating. And so basically you compare the stable isotope values of tissue from the predator to those of the prey, and then you can figure out what these animals are eating. I specifically look at one species, Neogonodactylus breedeni, right here. It's about 1 inch long, so this is not to scale. And it's one of the smaller species, but it does produce those amazingly fast movements. And where I work, and at Smithsonian Tropical Research Institute, this is actually on the Caribbean side of the Panama Canal, and the Panama Canal is about 3 miles behind me. If you're looking at this view in Galeta, then the Panama Canal is behind you. And so, and it's a really unique place to work because we have coral rubble and seagrass all in the same kind of habitat. They're sort of next to each other. And so you can, and these replicate plots, which is really interesting. So we can collect animals from these 2 different habitats and get a really comprehensive idea of what they're eating in nature, which is hard to do.
Is there a healthy coral further up?
Yeah, so this is a reef flat, I should say, and there's a lot of wave impact, and that's why it's a coral rubble habitat. The wall of the reef is here, and it's actually not great. It's not in the best. Yeah, it experiences a lot of wave activity, but it also does experience a lot of pollution and human interference. So it's certainly, there are nicer reefs to dive on. It's kind of what's become, party out there. Interesting. Hope you all can't, I don't know if you can hear the band music. Yeah, it's become kind of emblematic of Caribbean reefs today. So many Caribbean reefs are not, they look a lot like this, and they're actually not super healthy. So that's where I work. Mantis shrimp love it though. Mantis shrimp do really well in this coral rubble habitat where reefs are not necessarily thriving. which is interesting. And so we have, so here I'm showing you the stable isotope values of the mantis shrimp and their prey. And usually, if you're not used to looking at these plots, we plot carbon on the x-axis and the stable isotope values of nitrogen on the y-axis.
Keep going.
Oh, thanks. I wonder if can you guys hear it? I can't read the chat, but because of how this is set up, I don't know. So we have the mean prey items, stable isotope values of nitrogen and carbon, the mean values for the prey items, and then we have all the individual mantis shrimp that I sampled in the dots in both coral revel and seagrass habitats. And you can kind of look at these plots, and you can kind of guess what these animals are going to consume primarily. But you can also use excellent statistical models, specifically Bayesian mixing models, to understand the proportional contribution of each prey item to the diet. And for those of you who are really interested in Bayesian mixing models, I'm not including any of that here, but I love talking about it. So if any of you would like to talk about it further, I'm excited. I ran these in a number of different ways, and they all kind of showed me the similar result, which was this, that, so here we have all of the prey items on the x-axis, and then we have the median proportion of the diet of each of the prey items. And I think, obviously, they seem to eat a lot of clams, but they also seem to eat a lot of fish, which was surprising based on the literature. So remember that we thought that smashers were probably going to be specialists on hard-shelled prey, but here we have an animal where it looks like their main, or one of the things they really like to eat, are fish, which is a soft-bodied evasive prey item, which is something we thought that spears mostly ate. And I think that they're eating a lot of both of these. I think they eat a lot of fish because fish are super proteinaceous. They're actually easy for them to eat once they can get them. And these animals move their appendages really fast. So they don't have all of the cool morphology and the spines for capturing prey. But they're able to move fast enough to either smash it or spear it with the tip of their appendage. And I think they're eating a lot of clams because clams are they live very closely, if not on the same coral revel pieces that these animals live in. So I think they're just really accessible and easy to get. They're also pretty easy to open and smash. So that's my guess of why these animals are consuming these prey items primarily. And that's something that I'm really excited to understand more fully at this point. It's mostly just hypotheses, and I really want to understand, okay, how are they eating these animals in nature? So Breed and I lives on its own. It's one of a few smasher species that live in this habitat. But what happens if it's in the same habitat as a spear? Does that change its diet? So here's another place where I've worked, the Gump South Pacific Research Station run by UC Berkeley in Moorea, French Polynesia. And my field site is right here in front of the station. And again, if you were to put on a mask, go for a snorkel, you would see a lot of coral revel and kind of coral bommies like this. And you'd see probably a lot of little mantis shrimp eyes peeking out of these coral revel pieces. And this is an amazing place because it is one of the few places in the world where you find smashers and spears occupying the same habitat. So they actually live together in the same habitat. Both spears and smashers occupy cavities, the coral revel crevices, which is surprising because, again, usually you see spears in soft substrate habitats. But this is a really exciting natural experiment to be able to compare a spear and a smasher diet with where these animals have the same access to prey and where they live in the same kind of environment as well. So these animals are really small. They're the smaller guys. And then we have the smasher species and the spear species. And so here again, we have, this time I've plotted carbon on the x-axis, nitrogen on the y-axis, and we have spears in yellow and smashers in blue. And then we have all of the mean prey values, prey items as well. And then we see the proportional contributions to the diet over here after running the Bayesian mixing models. And again, clams seem to be really important for smashers. And spears seem to eat them too, but spears really seem to like fish. So when you put spears and smashers together, possibly we see a situation where smashers are kind of forced to eat hard-shelled prey, and spears because spears are better at eating the soft-bodied, more desirable fish prey. And so this is a question that I'm really excited to continue to pursue because again, these are different species, different parts of the world, but they're all related. And there are 500 species of mantis shrimp. So really we need to look at this in a phylogenetic context. And we need to better understand these diet morphology relationships. But I think what we're starting to learn is that spears likely competitively exclude smashers from consuming hard-shelled, soft-bodied prey. And
so the bar showed that the spears are consuming maybe 20% hard-shelled clams.
yeah.
Okay. So how are they even getting in there?
Again, the clams live in this, like, on the same burrows, that they kind of attach to the same cavities that these animals do. And spears can smash. So yeah, sorry, they can do it. They're not nearly as forceful, and it takes them a lot longer. But again, clams are much easier to consume than a snail, for example. Yes, these are good questions. And kind of along those lines, you can take these numbers and look at what's called a specialization index. So you can kind of compare how specialized they are on a continuous kind of metric that is the same, regardless of what they're actually eating. It's more just like, okay, how specialized are they? And spears come out as more generalized and less specialized than smashers, which I think is really neat. And again, more reasons why I'm excited to get back to Maria and understand this system better.
Is another way to say it opportunistic?
I think all mantis shrimp are opportunistic, way more than we originally thought. Yes, that is very true. That is definitely a good way to put it. So they have, these animals have morphological specializations, both spears and smashers, and that allows them to consume otherwise inaccessible prey. So smashers can eat hard-shelled prey when soft-bodied prey is not available. Spears can too. The reverse, they can eat hard-shelled prey when soft-bodied prey is not available, and they're really good at consuming soft-bodied prey. And I think, again, that this is likely a result of consistent competition for prey. Okay, so now we understand a little bit more about feeding interactions, especially when we think about competitive interactions as well, and then how this kind of fits into broader trophic dynamics. Of course, we've mostly looked down here. We haven't looked so much at who's eating mantis shrimp. That's also for future work. But I also really want to know how environment influences these feeding interactions, and then how that can influence trophic dynamics as well. So what I have become really interested in is ocean acidification. And we know that ocean acidification can affect calcified structures. We also know that the ocean is becoming more acidic with climate change. So the more CO2 we pump into the atmosphere, the more carbon dioxide the ocean absorbs, and that lowers the pH of the water. The ocean becomes more acidic, and that affects calcified structures. So basically, the structures can like literally dissolve in the water, but also what's really happening is that there's less calcium carbonate available in the water for these animals to build calcified structures. And that's very common what we see with foraminifera, corals, mollusks, like these snails, clams, and nautilus. Crustaceans, which are over here, are a little bit different. Crustaceans can show the response we would expect that I just described, but they can also show no response to ocean acidification conditions in the lab, or they can show sometimes even like a more robust and kind of thickening of their like a more calcified, thicker exoskeleton. And so it's really interesting to think about what crustaceans are doing, especially, of course, mantis shrimp, because mantis shrimp, I alluded to this, but they produce their insanely fast movements because their exoskeleton behaves like a spring. So you would think if they're going to be affected by pH, a lower pH and an increase in temperature with global climate change and the warming of the ocean and the ocean acidification, that's really could affect how their appendage behaves like a string. It could affect the structure, mineral composition, material properties, and that could then influence what these animals are eating in the habitats where they live. And so again, I looked at Neogonodactylus breedeni for this work. And this was back at Scripps Institution of Oceanography. We had 3 treatments. We had low pH, high temperature, low pH, and an ambient treatment. And then we had mantis shrimp and all these little cups down here. So our header tanks flowed into the cups. And we left them to stew in these conditions for 3 months and 6 months. And then we looked at their appendage afterwards. We also did some physiological stress response analyses to see how stressed they were in these conditions. But I just wanted to show this because I'm really excited to build a similar system at San Jose State. I think when some of the moving and shifting happens around here, we'll have more aquarium space and I'll be able to do things like this here, which I'm really excited about. We are doing some of this at Moss Landing, which I'll talk about later too. So very briefly, we have kind of the take-home message results from this work. And so you can look at the amount of calcium in the exoskeleton, the amount of magnesium in the exoskeleton, because magnesium is also really important for building these calcified structures. And then you can do material properties tests where you look at the hardness and the stiffness of the exoskeleton. And basically we found higher magnesium, which means a different kind of calcium carbonate, basically in low pH treatments at 6 months. So these animals might be kind of shunting around elements differently in order to construct their exoskeleton in the presence of ocean acidification, but it didn't affect the material properties, which is really interesting. So these animals were able to maintain their exoskeleton in terms of the material properties. What we don't know and what we're analyzing now is whether or not this affects the strike, because it could also affect muscle physiology and things like that. So we have high-speed video of these animals from these conditions as well that we're analyzing to understand, regardless of the exoskeleton, was the strike affected. And so basically though, it seems like mantis shrimp are really tolerant to these conditions. In the physiological studies that we did, we like dropped some animals down to 7.3 and they were like, whatever, they didn't seem to care, which is really low. A pH of 7.3 is very low for animals generally. And so they might be fine, but the hard-shelled prey that they consume in the future that are predicted to break more easily in future ocean conditions may not be fine. They may have a pretty big influence on the trophic ecology of the system, simply because they're probably going to be winners in this new acidified ocean. So we think about often winners and losers in terms of climate change in the ocean. And I think these animals are probably going to be winners, and I think that could greatly affect the trophic ecology of the system. And so the kind of broader things that I think we've learned from this work is that specialized morphology broadens diet, and we see that in mantis shrimp. That's actually similar to the cichlid fish, which also are super specialized. They have like some species have, or one species has like a jaw that's highly specialized of eating the scales off of the left side of other fish, and yet the majority of its diet is actually like algae and plankton and other organisms. And then also more recent work to what I talked about before shows that Galapagos finches also have a broader diet, and that they only really need to specialize in times of starvation or a low abundance of their preferred prey. And so I think that's probably what's going on with mantis shrimp. And I think mantis shrimp are also really important trophic links between macroinvertebrates and other intertidal organisms, like these, because we know that they consume a bunch of different prey, and they're also consumed by a number of different predators like lionfish and groupers and octopuses and stingrays. So I think that they're probably really kind of key links in the food web, and I'm excited to look at that further too. Okay, so I think we understand that morphology and behavior can inform broader ecological theory. I think we're also learning that the trophic dynamics of cryptic organisms like mantis shrimp that usually you don't see because they're hiding in their burrows are important drivers of ecosystem function. And the environment is really important because you can have shifts in feeding abilities and trophic dynamics due to climate change. So briefly, I wanted to talk about, just taking the time, cryptic fauna as important drivers of ecosystem function. So, mantis shrimp live in this tiny little cavity, but of course there's like a bunch of tunnels and crevices all over the reef. And there's a bunch of animals living in them, and we don't even know what they are, a lot of them. So coral reefs are 25% of the world's biodiversity, but we've probably only described about 10% of the species living in a coral reef. And so there's a whole ecosystem inside the reef that we don't even know what's going on there. And so we're starting to explore that. So we're starting to figure out who lives inside cryptic habitats and what roles do they play. So here's one of my grad students, Anna Rothstein, sticking a GoPro inside coral crevices. And this is what it looks like inside there. And here is a GoPro also with a pH and temperature sensor that we stick in the reef. And this is all in Maui. And basically, Anna and I have done a quick kind of analysis of the literature. And so based on kind of, a number of publications about cryptic habitats, we've seen kind of an increased interest in this topic. But most of it is about who lives inside reefs, and there's very little on the ecology and what's actually happening inside the reef. And so, but what do we know about crevices and the ecology of crevices based on this literature is that there's a lot of turf algae, which is this gray, brown, fuzzy stuff. And there's also calcified coralline algae, which we call CCA, and it looks like this Pepto-Bismol type stuff. And it's really important for cementing reefs together. And so Anna is really interested in how much CCA is inside of reefs that's kind of helping cement the reef together versus how much is outside of the reef. And then sponges are, of course, also really important for pumping nutrients and also making nutrients available for other organisms in the reef. So we kind of look at turf, CCA, and sponges in the reef right now. And so with an amazing group of wonderful undergrads who are all now graduated here at San Jose State, we looked at a bunch of this GoPro video, and we found that most of what you see inside a crevice is turf, but you do see a lot of CCA. And these are 14 different sites across Maui where I stuck a GoPro in reefs. And sponges are pretty low in abundance. But it gets kind of interesting because Anna went back to Maui in last January and also in August. And she, unlike me, turned her camera around in many different directions. And she found that the crevice wall looks a lot different than the top of the crevice or the crevice roof. So if you stick your GoPro in, if you turn it to look at the roof, we're seeing a lot more sponges and CCA than you do on the wall. And that's basically what this is showing here. We also looked at Peysonalia, which is another algae, which we're still figuring out why that's important. And also Anna is working to use machine learning techniques to quantify the percent CCA cover. And this is in very close collaboration, in fact, mostly doing the work of Dr. Phil Heller in computer science. So Anna is analyzing the videos with our techniques where we like literally point and click on dots and that in the computer and then we can calculate percent cover from that. But Phil has come up with this amazing automated way to tell us the percent cover of CCA in a photo, which is what's going on here. It's basically colored all the CCA patches by itself, the computer has, I guess. And that's going to be a way more accurate way for us to do this. And it's going to create, it's not going to be as much manpower, not take as many hours. So we're really excited about this. And then Anna is also comparing the distribution and abundance of key groups, the groups that we talked about inside the reef and the exposed reef. So we can basically quantify everything that I talked about outside of the reef and inside the reef and compare the two. And this was her amazing team in Maui in January 2022. And here is Dr. Phil in the middle. So he probably looks familiar. Yes, and I didn't get to go, but we're going back this January, so I'm very excited. Yeah, and it's just a wonderful team. Sophie Lou, a wonderful undergrad, got to come with us, and then Moss Landing, and then we also have collaborators at Scripps Oceanography, and one of the grad students got to come with us too, and it was just apparently a wonderful trip. So that is kind of the basics of what's happening inside the reef, but I'm really excited to look at these questions in kelp forests as well. A quick kind of ecology 101 on kelp forests is that they're here in California. They're super important and critical for California marine ecosystems. And usually we think of them looking like this, like these big, beautiful forests, the redwoods of the sea, not as long-lived, but certainly very tall. But sometimes, for example, very recently, it looks a lot more like this urchin barren. So this is actually the exact same site as this picture. And it was taken, I think it was a year later. And now, so kind of usually we see dynamics where you have kelp forests kind of flipping back and phase shifting to urchin barrens and then phase shifting back. But since kind of 2013, 2014, with disease and the warm water blob, I don't know if any of you remember that, but there was a huge warm water blob that came in and kind of hung around California. It destroyed the kelp forests. And also these urchins came in, were able to come in and just chomp down all of the kelp forests. And so now they're slowly starting to come back. This is a really important question in California right now of how do we restore our kelp forests? And so how are urchins really good at decimating kelp forests? Well, they're really good at reproducing, but also they have amazing jaw morphology. So this is their 5-part jaw that they use to, cut down kelp forests, but also scrape algae off of rocks. And sea urchins also are thought to have highly specialized morphology, like you see here, but they consume many different things as well. They love kelp, but they will scrape algae off of rocks and eat, even, in other urchin species, you find like little macroinvertebrates in their guts and things like that. So I'm really curious how this calcified structure of the jaw is going to respond to future conditions. And we know that when these animals are starved, they're able to maintain their Aristotle's lantern or their jaws. How is that going to change with ocean acidification? Are they going to actually be impacted? So people haven't really looked at their calcified structures and the impacts of that on from ocean acidification. And that's really important if we want to think about how resilient our kelp forest is going to be in our future ocean. And Sam Stein is my former graduate student. She just graduated last spring. And then Ryan Halsey is starting to continue to look at this question of this urchin kelp forest dynamics and how their eating influences kelp forest ecology. So briefly, Sam, in collaboration with wonderful undergrads, and we have a collaborator at California Department of Fish and Wildlife, and also Reef Check, which is a citizen science group. So Sam looked at jaw morphology in a kelp forest, but then also in 2 urchin barrens, and she compared kind of jaw length to test diameter ratio. So she measured, she took photos of the jaws. Basically, Reef Check sends us a bunch of jaws that they collect from all of these sites. And then we process the jaws, and then we measure the length of the jaw, the width, the curved length, and the area. Right now, we're really interested in the length because that's something that people have looked at in the literature since the 50s. And you can look at jaw length over test diameter as kind of to understand how jaw morphology might be changing in kelp forests and urchin barrens. And she basically found that you have longer relative jaw lengths. Sorry, the x-axis down here is test length and then jaw length. I know you guys can't see it. We have relative jaw lengths, and we looked at that in the summer and the spring of 2021. And the jaw lengths are larger in urchin barrens in the summer versus the spring, probably because nutrients are less available. But then that changes in a kelp forest. So in a kelp forest, we have smaller jaw lengths in the presence of kelp, probably because in the summer, kelp forests are doing a little bit better. Yeah, so I should say generally what people find in the literature is that you have smaller jaws in the presence of kelp. So this is consistent with that. And the reason why we think we see this seasonal change is because waters are calmer. Often in the spring, kelp is kind of pulled out of the water because of increased storms, but we have less of that in the summer. So now Ryan is really excited to learn more about how jaw morphology changes temporally and with kelp color cover, but also latitudinally and with ocean acidification. And so this is kind of twofold, but basically Reef Check has a number of different sites, all of them down California. So we're trying to get samples from all of those sites to understand this kelp forest urchin jaw dynamic along California. But then also we have upwelling. So every spring we have, the intensity can vary, but we have upwelling, which brings cool, acidified, nutrient-rich waters to the surface. And that's really important for our kelp forests. But also it kind of can be a proxy for ocean acidification because it's really acidified water. And we can look at how we can compare urchin jaws during the spring, during upwelling times to other times to see how ocean acidification might be affecting the structure. Eventually, we want to do, of course, ocean acidification experiments like what I did with mantis shrimp. We're just not quite there yet in terms of, yeah, we're just not quite there yet. So we're taking this approach of using upwelling as a proxy to start. So we can understand, again, this interplay between feeding interactions and trophic dynamics and environments by kind of looking at these broader oceanographic patterns and this really important key feeding interaction. And then really quickly, what's actually the most active part of my lab right now, but the part where I have the least data, is this project looking at the co-culture of abalone and seaweeds and whether or not when you grow seaweeds and abalone together, can you ameliorate those negative physiological and biomechanical responses to ocean acidification. So abalone are super important also in kelp forests. They actually, in a healthy kelp forest, they're competing with urchins and they're competitively excluding them from kelp forests. So they're helping to keep urchin populations down. But of course, in an urchin barren, they are very outnumbered. And in fact, urchins consume them as well. So, and what you're seeing here in this graph is that basically since the 1940s, till now, all populations of abalone have crashed. And in fact, you cannot fish for commercially or recreationally. You cannot fish for abalone in California anymore. The fisheries are completely shut down. So we have a real problem where we need abalone, both in the kelp forest and also for us, because they're a really important traditional food source, but also like they're just a really culturally important food source for Californians, both native and non-native Californians. Yeah, basically what's so interesting when I talk to students, they're like, really, I had no idea abalone were a big deal here. And it's like, yeah, well, that's because they were pretty much all gone by the time you were born. And so we're trying to, when you grow seaweeds and abalone together, the thought is that seaweeds, because they uptake CO2 because they're photosynthesizing, they can buffer the seawater. And so we know that abalone grow better from past experiments by my wonderful collaborators, Scott Hamilton and Mike Graham down at Moss Landing. But does this also improve the mechanics of the shell? And so that's what we're working on. Yes. And so Noah Colander is my grad student. He's based at MLML at Moss Landing, but he's working on this. He's running this entirely huge food, amazing experiment that we have going on there. And then we have wonderful undergrads, Nee and Diana, who come and they help measure the animals. And again, so this is a healthy abalone under scanning electron microscopy, and this is an abalone under acidified conditions. So you can see a lot of differences in the shell. And this is previous research on European abalone, but we expect to find something similar in red abalone, which is what we're looking at. So again, we're growing seaweeds and abalone together in these huge tanks that allow us to tumble the seaweed and create a lovely environment for seaweeds and abalone. And then we're measuring abalone growth, stress response, and shell structure and function. Yeah, and so, right, again, we can look at these two habitats to understand these interactions better. And I think I will leave it there because pretty much at time. Thank you. Thank you all so much. I'm gonna stop sharing.
So I've got my mic on now. Hopefully people can hear me. It's question time. And let's see. I always like to ask one. I'm going to ask one. Oh yeah, this is what I was going to ask. So when you raise abalone in the tanks at the aquaculture facility, are they suited to go out into the population in the kelp forests?
That is really great. Great question. And we don't know. And that is, I bet you Scott Hamilton or Mike Graham would have a much better, or Luke Gardner would have a much better answer to this. My answer is that we don't know. And that is actually a future direction. It's kind of part of the big NOAA grant that we just got to work on this. But it's really where I would love to see this go. So restoration aquaculture, I think, is really important. And so right now, what I didn't say is we're working on the juvenile stage. So we just want to know if we can get these animals through the juvenile stage, which is kind of really critical. It's a critical stage for them, and it's when we see a lot of loss. And then, yeah, once they're kind of adults, are we going to be able to outplant them? And we would love to be able to do, after we see how this works, is to take the white abalone, which we talked about a little bit before the talk, but white abalone are were the first federally listed endangered invertebrates, and they're still endangered, and there's a huge restoration program for them. So we want to see if this technique will work for them to help outplant those later as well. But yeah, that's where I would love to see this go, in addition to improving our sustainable seafood as a resource, restoration aquaculture.
I'm going to ask one more, if that's okay. You showed the before and after of that kelp forest that had been decimated by the urchins. Will that forest recover?
That's the crazy thing. Trying to remember the dates exactly, but 2019, 2020, everyone was just depressed and saying, no, it's going to take years for kelp forests to come back. And especially on the North Coast, where we have a species of kelp that takes longer to come back and reestablish, things are looking better in Southern California, but here in Northern California, it's a little more difficult. Anyway, it's still not great out there, but it is surprisingly better, and kelp is growing back faster than we expected.
Okay.
So that's good news. And it takes a lot. It takes like a big storm and kind of sweeping away all those urchins. So there's actually space for kelp to resettle. Disease can also knock out urchins.
I heard there are collection programs now too, right?
Yes, and that's also really exciting and something that Scott and Mike are both really involved in as well. And that's who we're working with too. So Reef Check works with divers. Urchin, these people they used to dive for a whole bunch of things, and now there's nothing to dive for except for purple urchins. And so they're diving for purple urchins. They're processing them on shore, and a lot of them are trying to send them to places in California where they're actually ranching them up. So they're trying to make a fishery out of the gonads of these urchin-bearing urchins, which actually have very few gonads because they're starving. They're not making eggs.
Right.
They're trying to ranch them up to make that a viable food source as well.
Okay. So I have at least one hand up. It looks like Madeline Marins has her hand up. Madeline, do you want to unmute yourself? And
no, sorry, that was
ask
on
your question
accident.
yourself?
No, I don't actually have a question.
Oh, and I had
That was
my
an
audio
accident.
off.
Thanks,
So go ahead, Madeline.
Oh, go ahead, Madeline. It's
Oh,
good.
can you hear me?
Yeah.
Okay, actually didn't have a question. That was from, I think.
Oh, that was from before?
Yeah, but thanks.
Okay. Okay, and then there's an anonymous attendee who asks, sorry if you answered this already, but why are urchins decimating the kelp forest so much?
Oh, sorry, that wasn't clear. They're just really good at consuming kelp forests. And so, yeah, there's actually, I wanted to have time to show this, but I didn't. You can Google urchins and kelp forests, and there's an amazing BBC video, like a David Attenborough video that shows just how amazing they are at using their jaws to basically like chomp at the holdfasts, which is where kelp hold on to the substrate, rocks and that kind of thing. So they're really good at chomping holdfasts, and then like entire kelp holdfasts just float away. And so they're really good at chomping kelp, and they're also really good at reproducing. So they are just constantly reproducing. They're really good at staying alive in these urchin barrens as well, on very, very little food. And so they're able to keep their populations going, which also keeps kelp forests down. So kelp forests aren't able to recruit back because the urchin barrens just exist there.
Great. Okay, Hillary Hurst has her hand up. Hillary, you should be able to.
Oh, Hillary is what Michael is saying. I don't know if you can hear.
Can you hear me? I don't know if you can hear me.
Now we can.
Oh, okay. Yeah, what a cool talk. It's really, really fun to learn about your work. I was actually wondering, so interesting to hear about the different effects on the mantis shrimp prey. And do you think that there will be enough of the unaffected prey to maintain the mantis shrimp populations, or do you think it's going to eventually catch up to them?
Oh, interesting. And I hadn't thought about that, but I think that mantis shrimp are going to be just fine. I think that they can eat so many different things. You know, I think I still probably didn't look at their entire, you know, the entire number of diversity of prey that they're able to consume. So I think that I think they're going to be okay. But, you know, who knows? It's a really good question. And also, we don't really know how these animals, these unaffected animals, are they really going to be unaffected? You know, there's a lot of research also on like fish behavior and the influence of ocean acidification on fish behavior, and like predator escape response and that kind of thing. So, yeah, these are all still really big questions. Thanks.
Thanks.
All right. I'm going to offer up one last chance for questions, but also want to respect that we're more than 5 minutes past the hour. and give people the opportunity to go do other stuff on a Friday afternoon. So seeing none, I don't think. Looks like we're all done. I want to thank Maya one more time for a great talk, inspiring a lot of questions, and look forward to hearing more about your work at Moss Landing and here in your lab over the coming years. Great.
Thank you all so much. It was so fun.
Thanks, everyone. Look for a notice for next week's. Oh, actually, I think next week we're off because it's Halloween costume contest, but I'll make sure to send out a notice about that. All right, good to see everyone. Have a good weekend.
Thank you.